Hi, thank you for joining me in this episode of GradSWE UCLA presents Women in Science, We Have the Power to Change the World. My name is Yifan, a PhD candidate from Civil and Environmental Engineering Department at UCLA. I'm also the International Student Event Officer at GradSWE UCLA. GradSWE stands for the Graduate Student Community of Society of Women Engineers. Our goal is to provide support to graduate students and help them shine in their future career. We are going to host a series of interviews to female scientists, and we hope we can encourage more women to join the community of STEM. Our guest today is Professor Smruthi Karthikeyan. She's Assistant Professor from Environmental Science and Engineering Department at Caltech. Her areas of interest are microbiome ecology, modeling of microbial dynamics to study ecosystem function, and so on. If you're interested in her and her research, please feel free to check her website. Hi, Professor Karthikeyan. It's really nice to have you here.
Thank you so much for having me. My name is Smruthi Karthikeyan. I just started as an assistant professor in the Engineering and Applied Science Division at Caltech. I just started last month. Before that, I was a postdoc in UCSD School of Medicine in Rob Knight's lab for about 2 years, and I got my PhD in 2020 from Georgia Tech. I majored in environmental engineering. I minored in microbiology, ecology, and computational biology. And I also have an undergrad degree in chemical engineering. I also have a master's degree in earth and environmental engineering from Columbia, just a year before my PhD.
Yeah, so what are you studying currently in your lab?
So my research interests are basically trying to link engineered systems like environmental engineering along with microbiology and computational biology, so we get a systems level view of ecosystems. Ecosystems can range anywhere from the environmental microbiome to the human microbiome. So I like using microbes to study much larger systems. So it's basically looking at much larger systems like the human health and disease, as well as the environment through the eyes of tiny microbes, because they're basically at the bottom of the structure. And any changes in the larger system are reflected in microbiome or microbial community shifts. And we can harness the power of these microorganisms anywhere from cleaning up, say, oil spills or bioremediation of other pollutants in the environment, looking at SARS-CoV-2 variants in the environment, all the way to how our human microbiome can predict disease. And you have a wide range that you can do by just looking at microbial systems. The one thing I do slightly differently is that since I look at environmental microbiomes, 95% of the environmental microbial diversity is uncultured, or they represent novel taxa or novel diversity. So typically when we look at microbial systems, we look at culturing, or we think, okay, to study microbe X, we need to grow them in the lab and see how they react to any changes in the environment. Well, that's very important. We also bias our knowledge to what can be cultured in the lab. And now when we do culture independent techniques like shotgun metagenomics, you're able to look at the entire, you get a snapshot of the entire community. If you take a snapshot of soil, there's millions and millions of different microbes, and the microbial diversity is enormous. And when you look at it in the lab, you're seeing maybe 1% of what is the actual representation. In order to make changes, for instance, if you want to know how this, how groundwater will be affected due to climate change, or how peatlands and permafrost will be affected with rising temperatures, you need to look at a system as a whole. So I like combining wet lab or experimental approaches with computational approaches, so you have a very complementary multidisciplinary view. So I like looking at the DNA, RNA, and metabolite level interactions in microbial communities, and I see if we can use the microbes as predictors for any environmental change or perturbation. And very similar techniques are also used to look at the human microbiome to see if there are any perturbations, to see, is this an indicator of, say, type 1 diabetes or any other disease? So, it really expands across human and environmental health, and they're very linked together too, right? Like, human health is very linked to environmental health and vice versa. So I like working on that interface.
Yeah, that's true. So that's especially like COVID-19. We all had like
Exactly.
3 years pandemic. And I know you're like famous for like COVID-19 variant sewage monitoring work. And I read your Nature paper, and you're also like featured in many media. You were nominated in the Science News like top 10 rising stars. So you just talk about like I read like your CVs and your introduction. It's like your PhD work, you didn't study like the COVID-19, but like we didn't have COVID-19 at that time. But you started with like biocatalyst, what I'm studying right now, and then like the microbial community in oil contaminated sites. So like, would you like to share like how your interests got involved into like COVID-19 sewage monitoring sequencing
Yeah, sure.
program? Yeah.
So I had a background in chemical engineering, so I had no microbiology or any biology background really. So I was very more into modeling flows. So I was in a very different field. And then when I switched to grad school at Georgia Tech, I started with molecular microbiology. My first advisor, Dr. Jim Spain, is a molecular microbiologist, does a lot of enzyme work and bioremediation work. So that's where I started. And so that's where I learned all the experimental skills. And then once he was going to retire, he recommended me to switch fields and learn more about microbial ecology and computational biology, which was very new to me. He said, it's a very good skill to have because it's very complementary to your background. So I switched to a different lab. Dr. Spain was still a co-advisor and a co-PI with my new PI, which was Costas. He does a lot of microbial ecology and computational biology work, and I was very new to that, but I found it very fascinating, especially because you can link environmental engineering and computational biology. So I was able to take what I learned before with bioremediation and then combine it with the ecology stuff, and I used that to use microbes to predict the impact of oil spills in marine ecosystems. And by looking at how we combine microbiology as well as, physicochemical parameters that are in the environment, you can link those two to actually predict how long, what the damage of ecosystems is due to oil spills at different scales. So it provides a very unique lens to look at a very traditional ecosystem, because you're not only looking at microbes in a vacuum, but you're actually placing them in context with the environment, because it's very important when you have, you have oxygen, nutrients, and other physicochemical characters, which are actually shaping your microbial community. So you need to see them together instead of looking at them separately, because only when you see them together, you get a more realistic view of ecosystems, right? So that's how I switched. After I got my PhD, I decided to move to UCSD, Rob Knight's lab, because they build all these tools for microbiome analysis. So I was hoping to actually integrate another dimension by looking at metatranscriptomics as well, and not just metagenomics. So I joined his lab to expand that to not only the gut microbiome, but also to environmental microbiomes, to see, can we include other data layers, like looking at metabolites and RNA transcripts, because DNA will tell us who's there, RNA will tell us what they're doing. So I thought maybe this would be a more integrated approach. Fortunately, I started my postdoc in March 2020, the same week as the pandemic. So I hadn't met any of my colleagues. So it was like a whole different, I had to do a complete 180 on my initial plans, because our lab was also a CLIA lab. And since we're in the medical school, we were also asked to pivot towards more pandemic research. So our lab was also processing nasal swab tests for the campus, as well as the San Diego County and California Department of Public Health. And at that point, UCSD decided to, keep campus open. And that time we were coming up with, can we, we're going to keep residences open. Is there an easier, cheaper way of testing everyone? Because nasal swabs, doing it every day for about 10 to 20,000 people is not feasible, because UCSD is a very large campus, right? Much like UCLA.
Yeah.
So that's when we were looking at wastewater, and a lot of people were already looking at wastewater at the community level to see if infections were going up or down. And we thought, is this something we could implement at the building level in the dorms? So we focused testing on only those buildings that actually have a wastewater positive, because doing it for one site across UCSD, even if someone's positive, is going to keep coming up positive, and we'd have to test everyone every day, and that just did not seem feasible.
Feasible, yeah.
Right. So UCSD is a large campus. We have about 350 campus buildings, and we had to come up with a strategy to be able to monitor wastewater every day, but also keep it at a way where it was cheap. And,
Mm-hmm.
we didn't need like 20, 30 people in the lab to be processing this, because processing wastewater every day is not an easy task. So that's when I came up with this robotic or automated system, which would process hundreds of samples in about a couple of hours. Because typically for campus to make decisions, we need the results the same day, or if you have a long turnaround time, that kind of beats the point. And CSD was like, we want the data the same day. So that's when we came up with a system where it automates the whole process, and it also sends emails to the residents automatically and builds a map, which anyone can go online and see which building was positive. And then we put vending machines in the buildings where people can swab themselves. If they're positive, they're moved to an isolation dorm. And that way the case counts were very low, campus was open, and the I think positivities were below 1% on campus. So this way we were able to keep campus open longer. And then we thought, let's push this further and let's see if we can do genomic surveillance, because, that's what is going to tell you what are the emerging variants. qPCR is very valuable, but you need to know your target, right? You need to know what mutations you're looking for. But a lot of times that means that you have to establish assays. And if you have a new variant introduced, it takes a while to establish assays and make these primers. So if you bypass that and do sequencing, you can know if there's a novel variant, you will know what are the new mutations, and if you can pick them up before. So that's when we came up with this method, which enable doing that, the experimental as well as the algorithmic computational method. We found that using that, you can predict these variants 2 weeks before. So that's what's currently implemented across the county and across other labs as well. And a lot of my postdoc went in that. However, I was still able to work on what I came to do, a lot of microbiome work. And after which I was at UCSD till December, and then I moved here and started in January.
Wow. So basically you can predict the new variant 2 weeks ahead.
Correct.
Wow. So basically the university can react based on the result at the same day and then make some policy.
Yes, and the county as well. So we also do it at San Diego County, so they can use that information to say, okay, we're going to see this variant. We've not seen it in our clinical data, but we're seeing it in wastewater. So it's only a matter of time before we start seeing it. So it also helps them focus their testings accordingly.
Wow, that's pretty cool. It's basically using the sewage will reduce this the swapping and also individual tasks and everything. Wow.
Especially in areas where resources are limited, right? Because
that's true.
So and it's cheaper.
Yeah, it's cheaper. Yeah. So right now UCLA is paying for students the qPCR test. We're using saliva, we're now using swapping. But still, it's it will be a huge cost for the university.
Exactly.
And there's only a few vending machines.
Right. And you have a large population too, right? And these days, most people are doing home testing. So you don't really know the true case. So this is just easier.
Easier for the
long-term
university.
Also.
Long-term monitoring. Yeah.
Exactly.
That's awesome. So you just said you didn't your original goal was not studying COVID-19, and then it suddenly changed. How was that feeling for you?
That was it was everything was put on overdrive, because everything was on a shorter time, right? Because of the pandemic, everything had to be done much faster. Traditionally in research, you have a time limit, but it's not so much, right? Everything. So our progress was exponential. You had to keep making these, let's say, innovations in a much shorter time than you typically do in research. So that was a challenge, but I think it's a very good learning experience too.
Yeah, especially you have a lot community experiences to support you, even you wasn't you didn't intend to study COVID-19 because you cannot predict it happens.
Correct.
But
Exactly.
So talking about the background, I noticed that you were minored in environmental microbiology, microecology, and computational biology during your PhD. So that's a lot of minors. I was we know PhD study requires a diverse skill sets, but but it's like minoring in different department might be like really hard. So, but I know there's like a lot of students like thinking about like minoring in different
Right.
departments. So could you please give some advice to those students, like our audience?
Yeah, sure. So since I came from a very different background, I did not have
Yeah.
any of this already, for research, but I really liked switching fields or sort of
Yeah.
doing interdisciplinary research because I find that the most effective when you try to look at the system from a different perspective. And I felt like this really gave me a new perspective and let me think in a very different way than I was conventionally used to. And I felt like I was very interested based on the research. I did take a couple of computational biology and ecology classes, and I was very fascinated and I wanted to learn more. So I took more of these classes so I would be able to appreciate it even more. However, it's not necessary that you need to take any of that, all of that, because as a part of your research, you end up learning anyway. So, and I think, a lot of people, I think the initial step is very scary, right? When you're switching fields, you always tend to go to something you're familiar because you're used to it. You think you'll be the most productive or you will contribute the most. And I think that is something a lot of PhD students are always worried about switching. They're more like, oh, I'm very used to this. This is my degree, so maybe I should continue. But I definitely recommend people definitely trying
Yeah.
other fields as well. It's the thought is more scary, but once you start getting involved and once you realize you really like it, it's definitely just a steep curve, but you can learn really fast. I don't think it's just the initial start that I think people are more hesitant, but once you get past that, it's definitely doable. It's not that hard.
The first step is like stepping
Yes,
out of
that's
the
the hard.
comfort
Yep.
zone. That's the hardest part. Yeah.
And it is going to take a little more effort, especially for me, because I was in a class with people who knew the background and I did not, but I really enjoyed that work and I knew how I could apply to research. So having a different perspective also helped, like you see with the SARS-CoV-2 research, having like an environmental engineering perspective and a very traditionally clinical background really helped bring a new dimension.
Yeah, it's like basically building up your background and will help your future work. I remember
Right.
when I was in like my master, I took two like microbiome ecology classes,
Mhm.
like two different departments. I was the only students in the engineering department. I basically don't know any terminology they are talking about, and I don't know like
That's
we know each
completely
other very
okay.
well.
Yeah.
Yeah,
It's
but I
completely
was like,
okay and expected
it's
too. So yeah, but it's
yeah,
worth it if that's what,
it worth it. Yeah,
yeah. So it's okay. So I definitely think if that's what you want to do, I don't think you should worry that your background was different. It's
yeah,
okay.
that's true. Yeah, let's like take a brave first step.
Exactly.
Yeah. So I think right now your brave first step is to being like a new faculty in Caltech. So you just mentioned you just started last
Mhm.
month. So it
Yes.
must be like pretty intense to build up like a new lab and to like have courses
Yeah, my lab's
open.
still being built. It's still
Yeah.
the physical lab is still being constructed. So that'll take a couple of months at least, but
Yeah.
it's good. It's a new part of a journey, so it's
Yeah.
exciting.
Do you have like any suggestion, any advice for like studying a new faculty position and any like interesting stories behind it?
I think so for me, I was, I would say first see where you want to be. So faculty position is like you have to be ready for it. It comes with a
Yeah.
lot, and that's not the only people should know that's not the only career
Yeah.
option. You should see if that's what you want to do. You shouldn't do it because that's what is expected. I feel like a lot of PhD students think that that's the only route. It's not. If that's what you want to do, then sure, I would say definitely pursue it. But it's always good to see if that's what you really want to do. If it is, then yes, I would definitely say, do your research before and see which of the universities, which of the departments are a good fit for you.
Yeah.
First, because that's a very important thing, right? Like, which is a good fit for you. So there, you can see what kind of research you want to do. Is it very specific to engineering, or do you want to do multidisciplinary research? In case you want to do multidisciplinary research, you have to see, would I be easily able to collaborate with others in the university? Because if you do want to, you have to see, oh, I have potential collaborators. So I would say definitely do a full thorough check of the department you want to apply to, see the background of all the professors who apply, because it's not always just your fit, right? It's always about who can I also work with to be successful, because ideally you want a very supportive environment. So I would say, look, yeah, exactly. Because when you're in, it's not like a vacuum, right? You'll be working with many different people. So I would definitely do my research before I start. So I used to go over all the faculty and their research in advance. And then I applied saying, oh, I think this is a very good fit for me because my expertise is in area X and I don't know much about area Y, but I think that might be helpful in my research. Then I'll see, oh, the faculty here is an expert in area Y. So maybe we can collaborate well together. So all of those are important too.
It's not like just like work individually or just like working with your members. It's like working with like a whole department or even like whole campus, not
exactly. And also you have to see, do you want a research focused role or do you want a teaching focused role? Because there are different positions too, where you can be, some people don't really want to write grants and are more interested in teaching, then they can take up the teaching role. But if you only like research, then you can take a research professor role. So it really depends on what, how much weight you want to give one over the other.
So it's like to select like a faculty position is one is like based on the interest, whether it's like you want to do research or not. And then like to see if there's like if you have the ability to collaborate with like people from different backgrounds, and then like building up the lab, I think probably you need to buy a lot of equipments and like work with different administrative.
Yes, you have to see which university is very supportive.
That's a lot of work, but I feel like it definitely, Intel Tech is always
to ask, and you have to be very, as a new faculty, you have to, I would recommend talking to new faculty in that university and see how their path to tenure is, right? Because you read like most of them will get tenure, but you also want to know if that process is if the university is very supportive of young faculty getting tenure, or if that process is very competitive or very hard. You ideally want to be in a place that is very encouraging, supportive of you getting tenure, and your tenure is only based on you and not other people as well. So that's very important, and that's something I would say is you have to keep in mind when you're applying, checking how the tenure process for each department or each university is. It varies very much, but you should see which one closely aligns
to your what you want actually for the department. So you started like to your PhD, your postdoc, you switched like university, even like met a pandemic and everything. So I have like following questions for my organization, the GradSWE, the Society of Women Engineers. So I wanted to also get to know you as a female scientist. So so my question is, as a female scientist, what challenges have you met in the way that you make these big decisions and like big change your life?
That's a very good question. So I think you would already know in engineering, women are very less, right? And it keeps reducing at each stage. When you start undergrad, it's like at least 40, 60, depending on the field, or even sometimes 50-50. But then when you go to grad school, it comes down a lot. And when you see in faculty, it keeps coming down even more. So, I think that's also because we have to make the whole field more supportive of women scientists and engineers. And in engineering, it's still very slow. It's definitely looking better than it was before, but I still think it's pretty, we still have a long way to go in making them feel more included, because a lot of times, let's say you're in a class and you're the only woman or female person in that class, you're a little more hesitant to answer because you don't want to be perceived as wrong. So all of those are, they may seem minor, but they shape your whole perception on whether you want to stay in academia or not, right? Because you want to feel more, you want to feel very comfortable in that space to be able to voice your opinions just as much as anyone else. So the more of us, it just makes it more easier for the next incoming set of women.
Like a supportive environment is really important.
Exactly.
So thank you. And then my next question is, do you have any female role model that you look up to as you were pursuing your career?
Actually, no. So that's a good point to have
Yeah.
more women in the field, because I didn't have enough women professors or enough faculty to look up to,
Yeah.
right? So I think this, this is a strong push for more women to take up STEM fields or engineering fields. So
Yeah.
there'll be more of us to look up to, and this will hopefully make the path easier
Yeah,
for
that's true.
becoming
It's, if
professors.
we have like more female
Mhm.
professors like during our undergrads, and like even like I think like when I was like searching for my PhD advisor, it's really hard to find like female
Exactly. Yep.
advisors in the fields. It's like, oh yeah. So I
Very few, right? You don't have too much of a choice too.
tried
So,
twice. Yeah.
and if that person's not in your field,
Yeah.
you can't work with them either. So yeah, so it's
Yeah, we need we
Mhm.
need to have like more female professors. Yeah. So my last question is,
Yeah.
do you have any advice that you would like to give, like, like all women who are thinking about or currently pursuing STEM field?
Yeah, like we just touched upon how representation matters.
Yeah.
Definitely, I would encourage more people to take that first step.
Yeah.
Even if it seems scary, but I think it'll definitely be worth it. So I would say if you're really interested in pursuing a certain field, and you don't think you have the background, I still think you should attempt it. It's
Yeah.
just the first step that's scary, but in the end, it's worth it. I can
Yeah.
tell you for a fact, I came from a background that's completely different, and I'm doing something completely different from what I started.
Yeah.
And it's okay if your interests change. The more you learn and the more exposure you get, you learn to like some fields more than the others, and that's completely okay. If initially you came into your PhD thinking this is what you want to do, it's completely okay to then change your mind and say, oh, I worked in this lab and I really think this is fascinating and I want to pursue. Completely fine.
Yeah. So it's to take like the brave first step. It's like
Yes,
really
yes.
important
Mhm.
for like all women, especially if they are just like thinking, like they are trying to
Yes, and I
decide
would
what they
say,
want.
yeah, and
Yeah.
have a very supportive and having a supportive environment really helps. So I had a very supportive, my thesis committee, my labs, my PIs are very supportive of the new ideas or directions I wanted to take. They didn't question it. They gave me the freedom. So I was also very fortunate to be able to do that.
Yeah, so the supporting environment is also
Yes,
like critical for,
yes.
yeah, for you, for like our audience to decide whether to pursue.
Definitely, definitely. I would choose a lab that I feel very comfortable in, and I feel like I feel like this is the environment that I could thrive in. So that's very important. I would definitely see how much I like the lab or work environment, because your productivity depends very much on that.
Yes, like a
It's not
happy,
just research. Yes, you have
yeah, happy life, happy work.
yes.
I would
You
say,
work better.
yeah,
You work better when you have less things to worry about. Yeah.
yeah, that's true. Yeah, totally
Mhm.
agree. Yeah, yeah. Thank you so much
Yeah, of
for
course.
all your insightful suggestions and the amazing introduction for our research. Yeah, thank you so much for today. Yeah,
Yeah, no problem. It was great talking to you. So
yeah,
well,
me
good
too. Yeah,
luck in your future endeavors.
thank you so much. Yeah, bye.
Bye bye.
Yeah. Thank you for joining me for today's episode of Women in Science. We have the power to change the world. Please hit the subscribe button and follow us to hear more stories from female scientists.